So I will switch to a slightly different topic related to brain function, which is the relationship between the information provided by the genome and brain function and potentially disease. We recently have been studying a phenomenon which I think is very intriguing and also potentially extremely important for brain function and disease, and that's the phenomenon of genomic imprinting, which we have studied recently in the adult and developing brain. The definition of genomic imprinting is the preferential expression of the maternal or the paternal copy of certain genes. All genes are present in two copies inherited from the maternal and the paternal genome, and this very intriguing phenomenon of genomic imprinting impose the silencing of some genes from one of the two parental genomes. In other words, some genes in your genome are expressed exclusively from the genome inherited from your mom or exclusively from the genome inherited from your dad. And that's this phenomenon of genomic imprinting. This is a phenomenon that has been studied so far mainly in the context of embryonic development. I must emphasize that this phenomenon is very recent evolutionary, which is that it emerged only in mammals, and I can discuss a little bit later what the evolutionary considerations are. In rodents and humans, they're thought to be about 80 to 100 imprinted genes so far. And in the context of the embryo, the idea is that the paternal genome is promoting embryonic growth, whereas the maternal genome is trying to restrict embryonic growth. And in fact, most of the paternally expressed genes, or PEG, are found to be growth promoters, and most of the maternally expressed genes are found to be growth repressors. So what about the brain? Well, a lot of imprinting defects that have been found both in humans and in the mouse also affect behavioral or neurological function. And the question is, how important is the phenomenon of genomic imprinting for the brain, and how involved it is in a number of mental disorders? One really interesting example of the effect of parental bias in expression and brain function and disease are found in this fascinating set of syndromes called Prader-Willi and Angelman syndromes that result from exactly the same mutation in the human genome, but in Prader-Willi, the inheritance is from the paternal genome, whereas in Angelman, the inheritance is from the maternal genome. So this is the cluster that is deleted in both syndromes, are indicated in red two genes that are exclusively expressed from the maternal genome, and in blue are a set of genes that are exclusively expressed from the paternal genome. Therefore, the deletion of this entire cluster will affect the expression of different genes. And the result is the following. In Prader-Willi, this cluster deletion is inherited from dad, and therefore none of the blue genes are expressed. And in Angelman, in which the inheritance is from the maternal genome, none of the red genes are expressed. And the phenotype of these two syndromes is quite striking. In Prader-Willi, the infants are hyperphagic, show impaired sensitivity to pain, frequent crying and tantrum, and psychosis. And in Angelman, it's almost the opposite, which is the infants show trusting personality, frequent loafer, and autistic features. And more generally, some authors have proposed that the involvement of this parental bias and imprinted loci might result in susceptibility to a number of mental disorders. So one question we were really interested in is, if indeed imprinting has something to do with brain function and in particular social behavior and homeostasis, then imprinted genes should be preferentially expressed in some areas of the brain. And so we use this fantastic resource that is the Paul Allen Brain Atlas to look at the expression pattern of all the known imprinted genes in about 120 adult brain regions. So this is an in silico analysis, which was just to take the list of all the non-imprinted genes and to look where they are expressed. And this is a sample of a set of genes and the expression pattern in 3 brain areas. As you can see, in some brain area, there are very few imprinted gene expressed, are represented, so each column represents a different gene. A gene that is known to be maternally expressed is represented in pink, and known to be paternally expressed is represented in blue. And as you can see, some brain areas have very few imprinted gene expressed, some have a moderate amount, and some have very high expression represented by these hotspots. If we do this at the scale of all the imprinted genes and almost all representative brain areas, we have something like this. Obviously, you can read, but let me walk through this. What you have here in green are biallelic genes that were randomly picked. And here are various groups of brain regions. And this control biallelic gene show us that there is a very high molecular complexity of biallelic genes in areas like the cortex, where there is known to be a lot of cellular complexity as well. So these are the hotspots for biallelic gene expression. So there's a lot of gene expression there. But if you look at the imprinted gene, here maternally expressed, blue paternally expressed, a lot of them are ubiquitously expressed, but some of them are expressed in very restricted area. And if you start to look at what are the hotspot or cold spot of imprinted gene expression, you can immediately see that they are very different from the hotspot of biallelic gene expression. In other words, there's something special about the brain areas in which imprinted gene expression occur. And if we look at the nature of these hotspots for imprinted gene expression, they correspond to areas involved in social behavior, motivated behavior, homeostasis, a lot of areas in the amygdala, in the hypothalamus, a lot of behavior that we are quite interested in, such as mating, maternal behavior, pain sensation, stress, reward, feeding behavior, etc. So this really triggered our interest in, what would the phenomenon of genomic imprinting do to the brain, and what specific function in the brain could this affect? So the question is, are there other genes that are imprinted in the brain, and in what brain areas? Now, how to start this? Well, there is a very intriguing experiment that pointed our attention to 3 different brain areas as really interesting target to look at genomic imprinting. And this is an experiment done many years ago by the group of Barry-Kevern that used manipulation in zygotes in order to generate embryonic cells that had a duplication of the paternal genome or a duplication of the maternal genome. These are called andronautes and parthenotes. Now, these cells, this zygote with a duplication of one of the parental genome, are unable to survive. But if you mingle them with normal embryonic cells, then these chimeric embryos are able to grow, to thrive. And one can look at the contribution of the paternal versus the maternal genome in the formation and the function of the brain. And what these results show is that there are different maternal and paternal contributions to brain structure. And I think these results are very striking, extremely interesting, and let me summarize them right now. So this is a normal embryo made out of cells with each maternal and paternal genome contribution. But if you generate a chimera with cells with a duplication of the paternal genome, then these embryos are very striking. They have a very large body, and they have a small brain. In contrast, if you generate chimeric embryos with cells with a duplication of the maternal genome, these embryos have a small body and a large brain. And this obviously suggests that there is something special about the contribution of the maternal genome to brain development. If you look later on, then something even more striking appears, which is that these cells with a duplication of the paternal or the maternal genome do not contribute to the same brain areas. Cells with a duplication of the paternal genome contribute exclusively to the hypothalamus, whereas cells with a duplication of the maternal genome contribute exclusively to the cortex. And these are the primary data here to show you in black are cells with a duplication of the paternal or the maternal genome. And you can see that there is a really black and white contribution to this brain structure. And so what that tells us, again, is that the maternal and the paternal genome participate differently to the development or maybe the function of different brain structures, and in particular the hypothalamus and the cortex. And therefore, we decided to undertake a genome-wide approach to try to identify all the repertoire of imprinted genes in 3 brain areas: the developing brain, the adult medial preoptic area in the hypothalamus, and the adult medial prefrontal cortex also in the adults. And so the strategy is as follows. We use a genetic trick to be able to recognize whether a gene was expressed from the maternal genome, the paternal genome, or both of them. And for this, we looked at gene expression in crosses, animals that result from the crosses of 2 very distant mouse strains, C57 Black 6 and Castaneus. There are a lot of polymorphism between these 2 mouse strains, and therefore every gene can be recognized as being transcribed from the paternal or the maternal genome. And we look at expression from F1 crosses and reciprocal crosses such that we can really distinguish what is the contribution of the species versus the contribution of the parent of origin. So the method was to perform high-throughput sequencing of the entire transcriptome of the preoptic area, the medial prefrontal cortex, and the E15 brain from the 2 parental strains, Castaneus and C57, and then the F1 hybrid and reciprocal hybrid. Our sequencing was very deep, 25 to 30-fold coverage of each transcriptome, and we have extremely good control of our accuracy in detecting parental bias. For example, over 90% of the SNPs from mitochondrial genes and 97% of X-linked genes in males are correctly called maternal. And we also can robustly identify non-imprinted genes, paternally expressed here, maternally expressed here. What you have here is the number of reads for a given SNP in one cross and the other cross. And this is mom and dad, dad and mom. And as you can see, this gene is exclusively expressed from C57 as being dad and Castaneus as being dad as well. This is a paternally expressed gene, and this is a maternally expressed gene. So what do we find? Well, the results are quite surprising. We found that from the original 80 to 100 non-imprinted genes, we've identified 13 genes with, that display imprinted features. About half of them are found in the UCSC database, so they are coding gene. About half are non-functional RNA or non-coding RNA. As you can see from the original 40 known and 93 known non-coding RNA, there's a huge expansion of the repertoire of imprinted genes. Importantly, the parental bias is very robust. We don't have some genes that are biallelic and then all the gradation to something that is monoallelic from the maternal or the paternal genome. What we found instead is that the mean parental bias is 90%, which would mean for a given read, 10 reads from mom and 90 reads from dad. So this is a very robust phenomenon, and we found it very reproducible across independent sample. We are not talking about random monoallelic expression. We are really talking about the differential contribution of the maternal or the paternal genome. And these genes are spread all around, all over the genome. Most of them are found in clusters, but some of them are found isolated. And so what this suggests is that the phenomenon of genomic imprinting is a major form of epigenetic regulation in the brain. This phenomenon of genomic imprinting is thought to be marked already at the level of the parental gametes, such that after fertilization, the cell always knows whether a gene originates from mom or originates from dad. And we think that these marks are actually being imposed upon a very, very large number of genes that are then expressed in the brain and that show this parental bias or parental silencing. One really interesting set of genes that we found is actually in the Prader-Willi cluster, such that there's this huge hole here, and we found 9 different transcripts, new transcripts here, that are all paternally expressed, and this is shown here by this paternal bias that is represented by these blue dots here, and this locus 4 that is found here show this extremely robust paternal bias with Illumina sequencing, and a PCR-based approach, Sequenome, also confirmed those data. Now, one of the most surprising features in our data is how these genes are spread between the embryonic brain, the adult hypothalamus, and adult cortex. As you can see, there are many more genes that show parental bias in the embryonic brain and fewer in the preoptic area in the medial prefrontal cortex. Interestingly, as predicted by the Chimenic experiment, you can see that there's a major contribution of the maternal genome at E15. There are twice as many maternally expressed genes than paternally expressed genes, and we found the opposite for the adult preoptic area and cortex, twice as many paternally expressed genes than maternally expressed genes. Now, interestingly, when we look at the nature of these genes, something very striking emerged. This showed that there's a major maternal influence on brain development and major paternal influence over adult brain function of offspring. Now, when we look at what these genes are and where are they imprinted, strikingly, most of them are found to be imprinted in only one brain area, only at E15 in green, or only in the preoptic area in blue, or only in the prefrontal cortex in brown. And this is despite the fact that 80% of these genes are actually expressed across the 3 brain areas, but they are found monoallelic in one brain area and biallelic in all the other brain areas. And so what these suggest is that the phenomenon of brain imprinting is spatially and temporally regulated. Now, I have to point out that these phenomenon, genes that would be monoallelic in certain tissue and biallelic in some other tissues, is nothing new. And in fact, the group of Mike Stryker very recently published a beautiful and very interesting paper on the switch of UB3A from being biallelic to monoallelic during the time of the critical period of plasticity in the visual system. But what we found is that instead of being an exception, it seems to be a general rule that the genes that show a monoallelic expression are actually monoallelic in only one tissue and biallelic in most of the other tissues. Now, one very striking example is IGF-2, which is a canonical example of monoallelic expression. These are the data. IGF-2 is here, H19 is here. You can see the expression data in black, and the parental bias in blue, paternal bias in red, maternal bias. Now, in the embryo, as expected, the IGF-2 gene is exclusively paternally expressed, and the non-coding RNA that is close by H19 is only maternally expressed, and only one of the 2 genes can be expressed. So this has been well described, and we found this data. But what is quite surprising is that in the adult, we found that only IGF-2 is expressed, and this time IGF-2 is exclusively maternally expressed, as you can see over here. So here we have a shift from paternal expression to maternal expression. And IGF-2 is a particularly interesting gene because it has been found its imprinting status has been found to be affected by starvation and stress in pregnant moms, such that the offspring of moms who have survived famines, for example, the Chinese famine or the Great Dutch famine, show a biallelic expression of IGF-2, and this is somehow correlated with high level of schizophrenia. Now, another very interesting set of genes is here, this cluster, dopamine decarboxylase, GRB10, which is a signaling molecule, cordon bleu, which is a gene involved in development. And you can see dopamine decarboxylase is maternally expressed. GRB10 is actually maternally expressed in the embryo and robustly paternally expressed in the adult brain. And these are the data here, the switch from maternal expression of GRB10 to paternal expression. Now, something also very curious that we discovered was the fact that some genes are actually imprinted only in males or only in females. And this is extraordinarily interesting in relationship with mental disorders, because as you may know, maybe most of the mental disorders have huge difference in the prevalence between males and females. So for example, eating disorders autism, schizophrenia are more abundant in one of the two sexes. And so if you have some genes that are monoallelic in one sex and biallelic in the other sex, obviously the sex that display monoallelic expression will have increased vulnerability. We found 150 sex-specific imprinted genes in the medial prefrontal cortex and 190 in the preoptic area. Here are the data, this very striking peak in the female preoptic area, where there's twice as many paternally expressed genes only in females than paternally expressed. And this is quite interesting, since in the female, the medial preoptic area is known to be involved in the regulation of maternal behavior. So we think that this differential paternal bias identified in male or female brain could really be lead to interesting candidates for sex susceptibility to disease. Now, finally, what about the X chromosome? All I've told you about it relates to the autosomes, and the X chromosome is actually particularly interesting as far as brain function is concerned. Although there are relatively few coding genes compared to the autosomes, the X chromosome has been shown to contain a higher ratio, the highest ratio of neural gene than any autosome. So there's something special about the X and brain function. Moreover, and correlate to this, it has been found that there are 3 times more cognitive impairment associated with the X-linked genes rather than autosomal genes. So what's happening with the X chromosome? Well, what we found overall, if we look globally at our data, something quite striking, which is that we found that globally, if we look at genes expressed from the maternal or from the paternal X chromosome, there are more, about 20% more genes that are expressed from the maternal X than the paternal X. Now, this is very curious, obviously, because one would expect that half of the cells in the cortex shut down the paternal X or the maternal X. And what we found instead is a preferential expression of genes from the maternal X chromosome. So we tried to look whether there was some type of bias in X inactivation that is found in the brain. And for this, we use a genetic tool, which is a mouse line in which a GFP reporter has been inserted under a CMV promoter in one of the X chromosome. And therefore, according to whether the transgene is inherited from the maternal or the paternal X, and whether the inactivated X express or does not express the GFP, one can recognize the cell by fluorescence. And if you look at the skin, the muscle, anything, you can visualize the expected random inactivation of the maternal and the paternal X, which is that there is this mosaic of fluorescent and non-fluorescent cell, about 50-50 for each of the X chromosome. But if we look at the cortex, again, if we look at GFP expressed by the maternal X or GFP expressed by the paternal X, what we found is that there is significantly more, about 30% more fluorescent cells that are expressing the maternal X rather than the paternal X chromosome. This is the prefrontal cortex, the sensory cortex. And we actually have looked at 5 different cortical regions, and we recognize this increased expression from the maternal X chromosome. But if we look in the hypothalamus, then we don't find this. We found 50-50. And so what this suggests is a very interesting and very intriguing phenomenon, such that mom seemed to be exerting an influence over the cortical function of the daughters through the X chromosome. So the sons all inherit the maternal X, so they get maternal influence here. But in daughters, when there is both the maternal and the paternal X chromosome, mom is trying to increase her influence by this preferential expression of the maternal X chromosome, which is a very intriguing phenomenon. Now, finally, the type of studies that we've done really bring a very new level of resolution to the phenomenon of genomic imprinting. In the past, people were looking whether a gene was globally expressed from the maternal or the paternal genome, but here our resolution is at the level of the SNP, so at multiple points along a given gene. So for example, this gene here was recognized by 5 different SNPs, and so at each of the SNP, we can do the read in both crosses, the F1 and reciprocal F1, and look at expression from C57 or castaneous parents. And this gene, for example, has clearly equal expression from the maternal and the paternal SNP copy in the genome at each of the SNPs. So this gene is clearly a biallelic gene. This gene is a clear maternally expressed gene, such that, again, in each of these 5 SNPs here, you can recognize there is a preferential read from the maternal SNP in each of the reciprocal cross and at each of the SNPs. So this is a clear maternally expressed gene. However, the majority of the genes actually that we found do not belong to that category, but belong to what we call a complex imprinting category, which is exemplified here. So what you have here is a gene that is represented by 4 different SNPs. And as you can see, the first 2 SNPs show preferential maternal expression, but surprisingly, the last 2 SNPs located here show preferential paternal expression. So what's happening? How can we explain this phenomenon? Well, at least for some genes, we can explain this phenomenon. And this one in particular, this is a gene called ARC3. And what we recognize is that the SNPs that were shown to be different than the other ones, so these 2 SNPs that display exclusive paternal expression, actually correspond to an exon that is uniquely found in a short transcript of that gene called ARC3. And so what we're saying is that this is a sign that this short transcript here is exclusively paternally expressed, and that this longer transcript here is likely to be maternally expressed. So the SNPs that are located here that are common between these 2 transcripts show that mix of both maternal and paternal expression, but this exon here that is unique to the short transcript enable us to uncover this preferential paternal expression of this short transcript. And we found many, many genes like this. This is an example of a complex locus, and there are relatively few of them, both among known and newly discovered imprinted genes. Cadherin 15 is paternally expressed all along the SNPs, and this is very robust. Other example of simple loci that we found is BCL2-like-1, which is a gene involved in apoptosis, and this gene with a complicated name, which is actually Argonaute2, involved in microRNA processing. This is a typical example of this isoform-specific imprinting, this gene H13. So just to help you localize yourself, H13 has 3 different transcripts, a short one, a slightly longer one, and a very long one. This is the expression data, and these are the parental bias. And as you can see, this long transcript here displays very robust and exclusive maternal expression. So this transcript is clearly maternally expressed. But if you look at the 5' UTR, the SNPs that are located in the 5' UTR of these 2 shorter transcripts, then you can see that they are exclusively paternally expressed. And interestingly, there is a gene insertion here in the middle that is also paternally expressed. So there are many other examples, very interesting examples of this complex phenomenon: astrotactin-1, interleukin-18, DDC, and TRPC9, a gene involved in mental retardation. And what that says, it's something that people have not thought about before, which is that the phenomenon of parental bias cannot be only investigated at the gene level, but now need to be investigated at the transcript level, which is within a gene, some transcript variant might be biallelic or show monoallelic expression from either the maternal or the paternal genome, but not all the transcript of the gene will follow the same rule. And so this is extremely intriguing and very difficult to understand functionally. But this is a more general question about what are different splicing variants doing in the brain. And Mike Greenberg was telling me that the BDNF gene has 48 different splicing variants. some are activity dependent, some are not activity dependent. Why does this gene need 48 different splicing variants? What's the distinct function or expression pattern of each of these splicing variants? Well, in addition to activity dependent, non-dependent, well, maybe also one need to look at whether they are preferentially expressed by the maternal or the paternal genome. So these really enter into the huge complexity of the phenomenon of transcription. So this really is the beginning of what I think is going to be a very important phenomenon in the brain, because the reality is, if genomic imprinting affects that many genes in the brain, and we look at just 3 brain areas. If we were looking at 3 more, maybe we would find 500 more. We really don't know. And what that suggests is that this is a very efficient mechanism to silence partially some of the genes. And this is a phenomenon that affects many, many genes. And I would argue this is a real phenomenon of epigenetic inheritance, because this is all due to epigenetic marks that are set at the level of the gametes and then are inherited in the embryo and in the adult. So there are a lot of questions that are raised. One is, well, what's the relationship between this imprintome and brain development and brain function? What's the role of all these imprinted genes? We've looked at brain areas, but the reality is, if there is this very specific regulation of genomic imprinting in specific brain areas in different sexes and during different stages of development, one will really need to look at the cell-specific basis, which is different type of neurons in a given brain area might have different imprintome. And finally, as I mentioned, different transcript in a given gene might also undergo different preferential bias. Now, the phenomenon of differential inactivation of the X chromosome is extremely interesting and intriguing. What are the different genetic programs that are unique to the maternal X versus the paternal X? And what regulates this phenomenon of the preferential selection of the maternal versus the paternal X only in specific brain regions? And then finally, something which I think is absolutely critical for the understanding of brain function and disease is the fact that these marks are set very early, in fact, in the gametes, in the primordial germ cells, in the pregnant mother. And therefore, one very interesting question is, how much is the environment, for example, diet or stress, are going to influence the setting of these imprinting marks, and therefore further on susceptibility to various disease. And I will just end here. The work was done by a terrific postdoc in the lab, Chris Gregg, helped by an undergraduate student, research computing, and this was a collaboration with David Haig in evolutionary biology. Thank you.